So we'll begin with the eye. I show this because the question is, how much of the brain do you think you use at any one time? And what happens if you were to use more than 10% of your brain? So the answer to 1A is not true. Okay, you become tired if you use more than 10% of your brain. Okay. Or C, you faint. So, yeah, raise your hands if you say yes to 1, yes to 2, yes to 3. The majority is right. The majority says yes, you faint. And it is the fact that you faint. In fact, if you use more than 1 or 2% of your neurons, you fire them at any together, you will faint, because you will use up all the oxygen that the brain can provide. Your brain is very demanding of oxygen, and the heart pumps 20% of the oxygen the lungs provide to the brain to keep it going. So our brains have evolved to be very, very efficient. This map that I have here in front of me uses 150 watts. Your brain, when it's going at full speed, only uses 20, So less than a single light bulb, So how does the brain compute all that it does with so few neurons? That's largely one of the questions that we'll be looking at throughout the course, find out why it's so efficient. And we'll start with the eye, because the eye is the key building block of the brain. So the eye is a window into the brain. The reason for that is it's actually part of your cortex. As a fetus, your cortex starts forming, and a bulge starts appearing from part of the cortex, and it grows out to become the eye. But if you look at the back of the eye, you see the neurons at the back of it, and they're actually, although they're in the eyeball and it rotates, it's not physically attached anymore to the brain, it has the neural structure of the brain. And so some of the new tests that are developing for Alzheimer's are, normally they look for plaques if you have Alzheimer's in the cortex. But unfortunately, those plaques you can only do with a biopsy from the brain. And that occurs either after you're dead, which is no use if you have Alzheimer's, or by a very invasive surgery. So developing tools just look at the eye itself microscopically with a very, very sharp lenses and look at those structure of the eye, the neurons, and test if you have Alzheimer's. So let's look at this fantastic structure called the eye. The eye consists of this thing, this big round thing. On the front, you have the cornea, which is one lens, and then you have this other lens behind it. And these two lenses focus the information out there onto the back of the eye, the retina. This is where all your neurons lie. And these neurons take the energy, the electrical activity, and send it down this optic nerve. That optic nerve then goes to your cortex. Now, this lens here, as I said, consists of this fixed part, which is your cornea. And then there's this flexible part that can change its shape. And you have here these muscles that contract, that can pull on this flexible part, and they encircle the eye like a donut. You can imagine this is a donut with little springs attached all along the edge around this sphere. So when these muscles contract, okay, this donut contracts, it becomes smaller in size, like diameter. It becomes thicker in its shape, but the diameter from one side to the other side of the donut becomes less. And when it becomes less, these springs become less taut, and the eyeball, or this lens here, wants to be round. If you let go of it, these springs let go, it becomes round in shape. So it seems a little bit counterintuitive that when the muscle relaxes, it pulls the lens to a flat shape. So you can see here in more than 3 dimensions how this works. Whenever the muscle contracts, the donut becomes smaller in diameter because these springs become less taut, and this lens here becomes more round. When it becomes more round, it focuses things that are near you onto the back of the eye. Now, people wear glasses or have other ways of changing their lenses. And 2 things can go wrong with focusing things on the back of the eye. You can have something wrong with the shape of the eye, either the eyeball being too long or too short, or this lens could be too flat or too round. When it's either too long or too round, you can have something that's called, you become nearsighted. So you can only see things clearly that are near you. You have trouble seeing things that are far away. And if that happens, then you would add glasses or lenses that are concave shaped. They are thinner in the middle and thicker at the edges. If your eyeball is too short, or the lens is too flat, then you're farsighted because you can see near but not far, or you can see far but not near, and you need convex lenses, and they're thicker in the middle and thinner at the edges. Now, if you get old like me, your lens becomes stiff. Okay, so this young lens can spring back to its normal round shape. This old lens, there's plenty of seats, yes, welcome, can't spring, so it remains too flat. And as a consequence, you have trouble reading something up close like a newspaper because you need the lens round to be able to see something close. Okay, so what type of lens do you think you would need in this circumstance? None, convex, or concave? We'll try. I don't have clickers here, so we'll try something different rather than raise your hands. If you could hit the table as hard as you can when I give it an answer that you think is right. Okay, all those that think none is the right answer. Concave is the right answer. Convex is the right answer. Okay, you got to hit it hard with energy. Try again. 1, 2, 3. Convex is Good. Well, I want to make sure it gets recorded so the students watching this know what answer it is. Yeah, you can see that with a convex lens it works. With a concave lens it becomes worse. Okay, the other thing that's very prominent in the eye is the pupil, or the iris. The iris is this thing around it. The pupil is the thing in the middle. It allows light to enter the eye. And when the iris contracts, the pupil becomes smaller. And it becomes smaller to prevent the receptors, which are very sensitive, from becoming saturated with too much light. But the other thing it does is, surprisingly, it also sharpens the way you see. Okay? You can see here the light's coming in, it goes through the lens, and here the pupil blocks it a bit. And in this case, you don't have the proper lens, so this object, this tiny spot here, gets focused behind the eye over here, and it's blurred on the retina itself. And you can see that when the pupil gets smaller, this blurring becomes smaller. Those things sharpen when the iris contracts. And this gives you sort of a more depth of focus too. You can, if you want to take, if you're using a camera, and you close down the lens, the aperture on the camera, your depth of focus becomes longer. You see things far away and close at the same time. Okay, what's at the back of the eye? Well, you've got 5 types of cells. The first one is these things called cones, which are called cones because their tips are cone-shaped. They're receptors, and their receptors, the cones, are of 3 types, and each type is sensitive to a different color. The other type is these rods, and they're called rods because they are rod-shaped. And they all are sensitive to every wavelength, so they don't differentiate between the different colors. And so they just see sort of in white and black and different shades of gray. The output of the eye, the retina, is this thing called the ganglion cell. And it's the thing whose axon goes down that optic nerve to the brain. In between, one of the cells we have is the bipolar cell. And that sends information collects information from these receptors. It sends it down to the ganglion cell. And then we have horizontal cells, and they connect the different receptors together so that they can go down these bipolar cells. In this case, I have these 2 connected to this bipolar cell. In real life, you would have hundreds of these receptors be connected onto a single bipolar cell. And so this bipolar cell, depending on how many of these receptors, these horizontal cells connect, have different widths, so it allows for different resolution, as we'll see in a moment. And then finally, the 5th cell is called an amacrine, and it does the same as the horizontal cell, but mostly for the peripheral, the rods on the peripheral side of the eye. Light has an interesting effect on these receptors. When you turn light on, the voltage, you can see here, drops. When the light goes off, the voltage rises. So it's as if dark is a stimulus to these retinal cells. Now, some cells, all these receptors, and then produce sort of these graded changes in activity. So one light goes on, the potential goes down. One light goes off, the potential goes up. Other cells, such as ganglion cells, and some of these amacrine cells, produce action potentials. And you've all taken action potentials last year. They are what allows information to transfer down a long axon to reach the brain. The rest of the neurons, these bipolar cells and these horizontal cells, are like these receptors. They only produce graded changes in potential. Now, why do they do this? Well, it's actually a handicap to produce action potentials. One of the main thing about this main handicap they have is it takes time for whatever cells receives information to respond to that change. So you can imagine this cell, a cell is firing, let's say, 100 times a second. And you can watch this screen to see all these action potentials. And it will take you 100 milliseconds, the distance between each of these action potentials, to decide if the next action potential is coming faster or slower. So the same thing, if it takes you 100 milliseconds to decide, so will the cell that's getting this information. So it's slower at making these changes. So it'll take 100 milliseconds then for the cell to start changing its activity downstream. Now, these graded change of potential are continuous and they're rapid. So all of this information gets transferred very quickly back and forth to all the cells that are connected. Now, so why do this switch from graded to action potentials? Well, it's because these graded changes can't travel a big distance. Now, that's not important if everything stays within the its retina itself, because everything's packed in there. But it does make a difference if you're going to send information all the way down to the cortex. So the ganglion cell must convert information to action potential. It has no choice. Okay. How many of you can read this? Maybe you can make out the title, London Free Press, but I doubt many of you can make out the small things. Okay? Now, so why is that? Why can't you read the newspaper in the dark? I can't even find the mouse. Whoa, press the wrong button. I'll have to quickly get here. All right. What's that? No, I've miscoded some of these things. Okay. So, it's difficult to read with low illumination for several reasons. First of all, the eye isn't uniform. The periphery here contains primarily rods, and the fovea here contains only cones. The other part of the problem is that these cones, in fact, are less sensitive to light, and whereas these rods over here are more sensitive to light. The third part of the answer is that the fovea, even though it's less sensitive light, it can see in more detail. And to prove that, if you stare at this diamond shape, many of you, if you don't take your fixation off, will not be able to make out the first word in the sentence is while. Many of you can't make out the second word of the sentence is staring. You probably might make out the third word is here. It's amazing if you stare at it, how little you can make out. And that's the reason you have to move your eyes from word to word to read. So, you combine all these reasons, and this is what you find. Okay? In the periphery, you have mostly rods, as you can see here, a few cones. But because they're all bigger, the spacing between each one is larger. Secondly, many of them come together and combine on a single ganglion cell. As a consequence, each ganglion cell sees a large part of the retina, but not in detail. So it has what's called low acuity. In contrast, the receptors in the fovea are these tiny little cones. So they have a high density, they're really closely spaced together. Only a few of them connect onto a ganglion cell. And so, because of the small spacing and low convergence, ganglion cells here see with a very high acuity. And as a consequence, then, what the eye sees by daylight is the fovea sees very clearly and in color. In the dark, the peripheral retina sees things more acutely, but at very low resolution and in black and white. The fovea itself is blind. Okay. So the optic nerve is small. Okay. So it's a bottleneck. It has to take all this information from this huge retina and condense it. And so, in fact, the fovea, which is about sees about 2 degrees and about 1% of the visual field, takes up about 50% of the cell axons in this optic nerve. So it somehow condenses a lot of information. It does so by a concept called a receptive field. How do you measure a receptive field? Well, for example, in this case, let's look at the receptive field of this ganglion cell. So the first thing we have to do is record its activity. So somehow we place an electrode near this ganglion cell and measure its action potentials. Then we take a stimulus, in this case, this appropriate stimulus for these receptors is light, and we pass them over the retina. So if we move it over to here, we find that nothing happens. The firing rate isn't changed. So we know that this receptor is not connected to this ganglion cell. We then move over to this one, and we notice that its activity becomes lower. So the definition of receptive field is that the activity changes. It can go up or down. That means that this receptor is in the receptive field of this ganglion cell. The same thing with this one here. It causes the activity to rise. And the next one here, it causes this activity to fall. So these three receptors are in the cell's receptive field, and these two are not. So we've defined the receptive field and what activity these receptors do. So the same definition applies to all visual cells, not just the visual cells in the eye, but as we see later in the visual cortex. And we use these receptor fields, we examine these receptor fields to determine what the function of these connections are. These connections serve an important function. And this same definition of receptor field applies to all the modalities. So you can think of a neuron that's getting activity from the skin in your hand, and you can plot its receptor field by, again, recording from that neuron in your skin, or a neuron upstream from that neuron, somewhere, let's say, in your spinal cord or your cortex, and using touch as the stimulus and defining its receptor field rather than light. A dead mouse. You have two of them and they're both dead. There's something wrong with the Bluetooth on this computer. It goes on and then off. Okay, the receptive fields in the ganglion cells, come in two flavors. There's ones that have a positive response, that is, you get more action potentials from them, more frequent action potentials, from an area in the center, surrounded by a halo that produces less activity from light in the surround. So these ones tell you when something's lighter. A good stimulus for this would be a big white light just over the center, not affecting the things in the surround. So, in contrast, there are these other cells that have the opposite. All the cells in the center produce less activity in the ganglion cells, whereas all the receptors in the surround produce more activity of the ganglion cell. So the best thing you could do for this cell is put a black dot here, not activating any of the receptors here in the center. And as a black dot in a white background. The white background then would activate all the surround, but not the center. So this cell senses that something is darker. This one lighter, this one darker. So let's look at the eye and pretend this mouse arrow is a light, source light. Okay? And this is a picture of what you'd see in the back of the eye were you to look into it. You would see these funny things, which are blood vessels, and they all come from the different parts of the eye and then congregate in this odd part here. And this is where your optic nerve lies. Okay. And this is then also where all the ganglion cells, axons, come and get together and then form the optic nerve and send the signal down to the cortex. This area in the middle here is sort of a little more red than the other areas, and that is where your fovea lies. So that's the most sensitive part of it. Okay, so let's now explore this with this spot of light, and we can see here that activity goes from this basal rate to this higher rate, and then drops here, becomes less than the basal rate, then goes back up again over here, and then back down to the basal rate. So there's an up, down, up. If we follow this around into a ring, you can see there's a ring of positive somewhere around there. And you can just plot your way. And after a while, you'll discover what the shape is. You'll notice that there's an area in the there's a negative area in the center, and that's located around this blood vessel here. Okay, if you look at this picture, this is what you get if you put dots where things change. Okay? And you can see here, it's an off-center, on-surround ganglion cell. Now, there's other cells. We've seen now only cells that turn light on, it either decreases or increases the activity. But you've also got these phasic cells. When you turn light on, it increases the activity, but then the activity gradually returns to baseline. And then you turn off the light, and it silences for a moment, and then goes back to baseline. So turning the light on produces large activity, then it dies, and then turning it off does the opposite, turns it off, and then it goes back on again. So these are called phasic cells. Now, how do you form these receptive fields? Well, let's suppose this was the center of one of these centers around ganglion cells, and this is in the surround area. And this is the ganglion cell we're talking about. And this is an on-center cell. Let's see why it becomes on-center. You can see that light goes on here, the voltage drops, that releases less transmitter. This is an inhibitory connection. Putting out less inhibitory transmitter allows the voltage here to rise. It then produces more activity because it's a positive connection to the next neuron, the ganglion cell, and if this rises, this rises as well. So turning light on here produces an increased activity in the ganglion cell. Now, in a cone in the periphery, a more complicated event happens. Okay? Here we have a positive connection, and here we have a negative connection. Okay? So what happens here is when light goes on, the voltage drops, less transmitter here, then more activity in this. If it's less inhibitory, the activity in here will rise. When this rises, this then will produce more inhibition. This will fall, and this will fall. So light here in the surround will produce less activity here in this ganglion cell. So that produces the inhibitory surround. An easy way to think about this, rather than less transmitter, more transmitter, is think of a positive connection means that whatever happens here as well. A negative connection, like this one here, whatever happens here, the opposite happens in the downstream neuron. So I think that's an easier way of looking at the problem. Now, why are we doing this? Okay. Well, it accentuates the edges. Let's see. Now, accentuating edges, artists discovered that's important a long, long time ago. You can see here that this is a darker gray, and gradually it lightens. Now, on the other side of this edge is a very dark black, and gradually that lightens. So as a consequence, this edge appears in more contrast than if it were the same color throughout. Like if this was the color of the blouse all the way, and this was the color of the background all the way. So artists like to accentuate edges. Well, the brain does the same thing. Neurons that are far away from the edge, let's suppose this is a white and this is dark, bright, dark. So these cells here, far away from the edge, are not affected because the excitation here and the inhibition here is balanced, and they continue firing at their low basal rates. The same thing happens for neurons over here. It's only the ones at the edge that change their activity. This on cell becomes more active, and this off cell over here becomes more active. The two types of cells together help the brain define that there's an edge here. Now, we asked the question a long time ago, how does the brain compute what it must with such few neurons? Well, this is one way. It just senses change. And so the number of active neurons are kept to minimum. In this case, the most active neurons are the ones at the edge. Now you can see here that this is a gray. This is the same color all the way through. This is a darker gray, the same color all the way through. A darker still, darker still. But if you look, you seem to see this. It seems to be lighter at this side and darker on this side. Same thing over here and same thing over here. These edges are being accentuated. And these edges are being accentuated by these ganglion cells. The other thing these ganglion cells do, by primarily concentrating on the change between at this edge, is produce constancy. If you take a piece of newspaper that you're reading and read it indoors, you see a black letter against a white background. You take the same, but because it's darker indoors, the amount of light reflected for both is much less compared to that when you see it outdoors. Both will reflect more. If you put the two together, you find a remarkable thing, that the black of the letter is actually brighter than the dark of the background. And yet you see things, the letter is black outdoors and the letter is black indoors. And that's because the change between here and here is the same as the change between here and here. The amount of the letter is darker by the same amount between the paper indoors and the paper outdoors. So it maintains the constancy. Okay, the last thing I want to cover is color vision. Now, these apples and bananas, the picture I took from the basket in my home, taste a lot better when you see them in color. You can see whether the apple is ripe, when that banana seems ripe, what kind of apple it is to differentiate it from the orange. And many people believe that we see in color in order to detect things like food that tastes better and is good for you. Now, we detect this color with these cones that I mentioned before. This one here, when light hits a cone, it activates the cone in different colors will activate the cone, some more than others. You can see here that this cone will be affected by all these colors, but primarily by the green here in the middle. Okay, so we call these green cones. Now, a green cone, when it sees this, has a problem. Okay? This is the color of the background, and that color is over here. where the letter A is. This is the color of the object in the foreground. That is how much stimulus that color will produce. That is about the same thing as that, and the consequence, you'll see this. Okay, not very good survival value, right? So what the brain does is have more detectors. So for example, if you had a green and a red cone, A would give this much stimulus, B would get this much stimulus, a lot more. And now this bear would be clearly seen in the background. And so many species, like us, have more, and we have 3 types of cones. And a few others have even more than 3. Now, there's still a problem, even with 3 cones. And we'll see the solution to the problem just a few minutes from now. The problem is this, okay? You can get an increase in how much stimulus, how much the receptor is activated by, either by 2 things. One is a change in color, so the color can go from here to here. That'll produce an increase in stimulus. But also, the brightness can go up, and it can go from here to here. So more activity, is it because it's brighter or because it's a different color? How does the brain differentiate between those 2 situations? And we'll see in a moment that's solved by a special cell in the cortex, visual cortex of the brain. But before we get to it, let's look at color in more detail. So we have 3 types of cones: red cones, green cones, and blue cones. They're not that color when you look at them, but they're more sensitive to those colors of light. Now, when you combine colors of light, it's very different from combining colors of paint. That's a very important thing. So when you combine green and red light, you get yellow light. You don't get that when you combine those colors of paint. When you combine all the colors of light, surprisingly, you get white. If you combined all the colors of paint, you get black. In your retina, you have these cones, and the distribution of these cones changes depending on where those cells lie. In the fovea, you have all 3, but red predominates. Green is less, and the least is blue. They also form clusters, like you can see this cluster of red, a small cluster. You can think of this as a cluster of green, and so forth. The other thing is, at the very center of the fovea, you're not sensitive to blue. And as you go to periphery, the number of cones decreases, and the number of rods starts appearing, and they become larger, both the cones and the rods. And so the density decreases, and the cones become larger than rods. Now, color blindness. Now, if you look at this thing, and you're not colorblind, you can perhaps see a 1 here and a 0 here. Okay. So your sensitivity to these different colors allow you to group them together and see these numbers. Now, if you can't see the number 10, don't worry too much. It might be that the screen colors on this that are being projected are not accurate. Now, you get color blindness for a variety of reasons. You can miss a certain receptor, either no red receptor, no green receptor, no blue receptor. You can miss two of the receptors. You can miss all three of the receptors, or the shape of this curve can be different from normal. Now, these occur because there's a photopigment in these receptors, and that photopigment is produced by your genes, and you could have a deficit in these genes. Now, let's try this test again about with the hand clapping or hand hitting the table as hard as you can. Now, suppose you lost all three cone receptors. If you lost all three, A, you couldn't see. No one clapped. You couldn't see a red apple. Okay, there's a few claps. You would see in dim light. You would not be able to see in dim light. No one. You could read. You couldn't read. Sorry. You would not be able to read. But try again. For you not be able to read. Those that say yes to that, hit the table and start again. Oh, that's a weak response. So my mouse. That is surprisingly the only correct response. Now, you wouldn't see, you probably may have thought that you couldn't see a red apple, that is, you could tell it's an apple, the redness of an apple. I didn't mean that. That you would be able to see a red apple, but you perhaps couldn't tell it was red or green or whatnot. You'd be able to see the object because you still got your cones. Okay. You'd be able to see, or still got your rods. You'd be able to see because you got your rods, but you couldn't tell it's red. You'd be able, you can see quite well with rods in dim light. And correct, you wouldn't be able to read because you need your cones to read. They're the ones in your fovea, and they're the only things in your fovea, and that is the place where you have your highest acuity. Okay, now you can see how many different shades or gradations of color can you see with just three cone types. Well, if you take all the different hues, I've sort of digitized them to a solid hue, but you can imagine there's a continuum from this purple through this orange, through these greens, and through this dark blue coming through it. If you would test people between being able to differentiate between this and its nearest neighbor, you could find that you could separate 200 different hues. If you then take these hues and combine them, so you take this green and this purplish color, you can combine them in 20 different levels. The middle level, of course, will appear white, but there will be different shades or saturations. Now, in this case, we're combining complementary shades. So this green lies in direct opposition, so through the middle of this shade of purplish. And so it produces white. If you take this green and this red and you combine this, you'll get this yellow. Finally, if we take different brightnesses, you can detect about 500 different brightnesses. These circles are smaller here and here because very bright colors you're less able to detect differences in very bright colors as you are of very dark colors. But if you multiply all these numbers together, you find that you can detect 2 million gradations of colors. And so any screen that can produce more than 2 million colors, computers advertise, well, we can produce, more than 2 million different colors. It's not of much use because the brain can't detect more than that. Okay, here's an interesting puzzle. Okay, and we'll be, the next part of this class will be trying to explain this phenomenon. Stare at the X, or plus sign. And if you stare hard enough without taking your eyes or looking at the sides or anything like that, try to ignore the thing going round and round, something remarkable would happen. The first thing that might happen is that you see the color of the thing that goes round and round starts turning to blue. And then if you keep fixating and not turning your eyes, something more remarkable will happen. That the yellow dots will disappear. So you have to be really concentrating on it. Okay? How many of you got the yellow dots to disappear? Ah, not bad. You're good at fixating. How many of you got a blue dot going round and round? Okay, almost everyone. Good. Okay. Why is that? Okay, everyone that thinks it's A, there is a rotating blue dot that appears late in the scene. Okay. Those that you think that there's a yellow dot, these dim late in the scene. Okay. C, those that think there's a yellow dot that briefly turns off and there is in fact no blue dot. Okay. Anyone know what the reason for that is? Yes. That's very good. Yes, there's adaptation occurring from the continuous yellow. And when that you adapt to that yellow, something remarkable happens. When you turn off that yellow, its opposite appears. So let's look at that more detail. If you were, just as an aside, if you stare at a particular fruit, something remarkable will happen in this scene. The other fruit, or all the fruit, will start appearing in color when the scene is in fact only in black and white. Okay? Again, because you have the same adaptation going on. Okay, let's try the same thing here. Stare at the X. And if you after a while, you should see halos appearing of all the colors when they go off. If you keep concentrating on the X, you'll see that a different color appears in each of the halos. Okay? You should see that the red circle leaves a cyan or pale blue after image. You see that the green leaves a purple magenta, or a purplish after image, and that the yellow leaves a blue after image. So it looks like these things are paired, that they're paired in opposites, This one leaves this as the after image. This leaves this as an after image. Similarly, these two are paired, and each one leaves its opposite as the after image. Why are they paired? They're paired because we have these special cells in the cerebral cortex that solve this problem of things either becoming brighter or changing color. Okay? It's able to detect which of these is happening. And the way it does this, by having this type of receptive field. For example, here, there would be a double opponent cell that's sensitive to blue light in the center and yellow light, which is a combination of these two colors in the center as well. But the effect on the center is a positive for blue and a negative for yellow. So it's called, that's one opponent. It's called a double opponent because in the surround, the opposite happens. Okay? Here, yellow inhibited. Here, yellow excites. Here, blue excites. Here, blue inhibits. We also find sort of a green-magenta double opponent cell. Now, that explanation that was given, what happens when you have a light that's continuously yellow? Gradually, the receptors that are sensitive to the yellow light adapt. Okay? They produce less and less activity to the yellow light. So now we turn off the yellow light, but replace it with white light. Okay? If we do that, white light contains all the colors, including the yellow. Okay? But all the colors include blue. So we activate these guys. That produces an activation of this double opponent cell. As a consequence, we see blue. So when the light goes off, and after adaptation, we see this blue light. So the after image is blue. And similarly, we see a different after image for other colors. Hurry down a bit so it doesn't take too long. So we can see the same thing happening here. Okay, all we have is this flickering, and this flickering means that this turns off, but it's replaced by gray, which is like white, but slightly less bright. Okay. And as a consequence, if you just stare at this carefully, you'll see that its after image is blue. Okay? So just by itself, staring at this yellow light, turning off, a blue light appears. And gradually, each of this dimming produces a yellow light, a blue light, and you see it rotating round and round. And if you stare long enough, you'll adapt your receptors for yellow so much that they'll disappear. Okay, what other things happen with these double opponent cells? Okay, what color will most activate these double opponent cells? Okay, if we try shining blue light on the double opponent cell, we see activity. Okay, if we turn yellow light on, oops, we'll go back one, forward one. Okay. Yellow light produces this I've misprogrammed this, so if I hit the wrong color at the wrong time, it doesn't go in the right sequence. So that's why I had to back up. Anyways, turn on the yellow light, it produces less activity. Turn on the blue light, it produces more activity. So what do we do to the surround to produce the most activity? Well, we turn on blue light, it reduces the activity. That's not good. So what if we tried yellow light? So you can see yellow light produces, in the surround, produces the biggest activity possible. So something blue in the center, surrounded by yellow, will produce lots of activity of the cell. If we switch the polarization around, then something like this tie, yellow tie, will appear very bright against this blue background. Okay? So I'm wearing this thing for a reason. You will not see me with the tie on for the rest of the course. I don't like wearing ties. The other thing that you have is a constancy. So if we shine light on both the center and the surround, you see no activity. Same thing what happens yellow light. Any color that we put on everything doesn't disturb the sensitivity of these cells. Okay? And so this produces a sense of constancy. They are not disturbed in their activity. They're just sensitive to changes in the color of light. Now this is different from cameras. Cameras are very sensitive to the color of light. You take a picture of a scene indoors with incandescent lights, not LEDs. You'll generally have yellowish hues to it. If you take it with the same setting outdoors, things will have a bluish hues. So cameras don't have the same color constancy that the eyes do. So just to end this thing, the eye does three things. First of all, it focuses light onto the back of the eye, on the retina. The retina then detects the various colors and intensities, but it compresses this information by concentrating on the changes. So it has to compress because it has a small optic nerve. If the optic nerve were as big as the retina, the eyeball won't be able to turn. You couldn't turn it to different points in the scene. Now, look. So this compression I mentioned occurs in two ways. The detailed image, only a small part of it is in detail. That coming from the fovea. And secondly, only the changes, either in color or brightness, are transmitted. And so many of these receptors converge their information on a few neurons in the cortex. So the result is a transformation. That's sort of the other title of this course, Transformations, Action, Perception. And what it seems to transform, this is what is there in this scene, and what the optic nerve sends out. You can see color from where the fovea is, and less information as you go towards the center. It seems like a poor representation of what we actually see, and yet we see it in a lot of detail. You know, what you're seeing now is very vivid to you, and how it becomes vivid will be what we'll concentrate on in the next few lectures. So, thank you very much, and we'll see you next week.